Good morning, children. Now, today we are going to see about the chapter 9, that is the unit of life. You know very well the unit of life is nothing but cell. Cell is the fundamental, structural, and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a living cell. Robert Brown later discovered the nucleus. The invention of the light microscope and its improvement leading to the electron microscope made it to reveal all the structural details of the cell. And the cell theory, it is proposed by Schleiden and Schwann, in which we can study cell structures or cell structures. So cell theory as understood today is all living organisms are composed of cells and products of cells. All cells arise from pre-existing cells. And also we have different shapes of the cells. For example, mycoplasms, the smallest cells, while bacteria would be 3 to 5 μm. And the largest isolated single cell is the egg of an ostrich. And also when they vary in different shapes, they may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular shapes. So now we are going to see some of the cells, cell organelles in this chapter and this video. So now we are going to see the prokaryotic cell. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma and PPLO, that is pleuropneumonia-like organisms, and they are generally smaller and multiply more rapidly than the eukaryotic cells. They may differentiate greatly in shape and size, and also some of the shapes of bacteria are also present. And now when we see the characteristics of a prokaryotic cell, cell is surrounded by a cell wall made up of lipids, proteins and polysaccharides. Nuclear membrane is absent. Single circular coiled naked DNA called nucleote is present. Nucleolus is absent. Mitochondria, plastids, lysosomes, peroxisomes and cytoskeleton is absent. And respiratory enzymes in cell membrane is present. And prokaryotic cell divide mitotically. And 70 type of ribosomes are present. So these are the 8 characteristics we can see in the prokaryotic cells. Now coming to the eukaryotic cells. The eukaryotic include all the protists, plants, animals and fungi. So in eukaryotic cells there is an extensive compartmentalization of cytoplasm through a presence of membrane bound organelles. And now we are going to see 2 cell organelles that are double membrane bound, and we are going to see its characteristics, its functions and of course the diagrams. So the 2 cell organelles that are double membrane bound structures are mitochondria and chloroplast. So now coming to the mitochondria, we know very well in a saucer shape we can see the mitochondria, and cylindrical or of course rod shaped also is found. It's a double membrane bound, outer and inner also, and the space between these 2 layers is perimitochondrial space, and the inner membrane forms infoldings called cristae. They increase surface area. Inner compartment is filled with matrix which contains enzymes for aerobic respirators. They produce cellular energy in the form of ATP, so they are called powerhouse of the cell. Matrix has DNA, RNA, ribosomes, that is only 70S, and enzymes, and they divide by fission. Now coming to the chloroplast. Chloroplast, it is one type of pigment in the plasmids. So plasmids are found in all plant cells and in euglenoids. These are easily observed under the microscope. Plasmids are nothing but plastids. They base some specific pigments. So based on this type of pigments, plastids can be classified into chloroplast. Now coming to the chloroplast. The double membrane, it is a double membrane bound, outer and inner also. And the space between these two membranes is called as periplastidial space. Flattened membranous sacs are called thylakoids. Thylakoids arranged as piles of coins and is called as grana. And thylakoids in grana are called grana thylakoids. Okay. And thylakoids in stroma are stroma thylakoids. And the stroma contains DNA, RNA, ribosomes, enzymes for photosynthesis. And here we can see the synthesis of carbohydrates by photosynthesis. So these are the few characteristics of the chloroplast. And now we are going to see about the centrosome. It's an organelle usually containing two cylindrical structures called centrioles. And now the cartwheel structure of centriole we are going to see. Two cylindrical structures called centrioles, centrosome, and surrounded by the pericentriolax materials, and centrioles in centrosome. They lie perpendicular to each other. They are made up of 9 evenly spaced peripheral fibrils of tubulin. Each fibrils is a triplet. Adjacent triplets are linked. Central part of the centriole is called hub. Of course, it is a proteinaceous. It is connected to tubules of triplets by proteinaceous radical spikes. So this is the cartwheel structure of the centriole. And now we are going to see the nucleosome and how are they made up of. So coming to the nucleosome, that is chromatin appears as beads of strings. These leads are called nucleosomes. It contains 200 BP, that is base pairs of DNA, double helix wrapped around a core histone octamer. Histone octamer contains 2 copies of 4 types of histone proteins. You can say H2A, H2B, H3 and H4. HI histone molecule seals the 2 turns of DNA by binding the point where DNA enters and leaves the core. The DNA between 2 nucleosomes is called linker DNA. So this is about the nucleosome and how are they made up of. And one more we are going to see in this chapter is plasmids, which is also of course important. Just a small question. So plasmids and what is their role in bacteria? So plasmids are small circular DNA molecules other than xenomic DNA are called plasmids, and they're present in bacteria. It gives some unique phenotypic characters, and these are resistant to antibiotics and transformation, etc. Okay. Thus we can say the cell is a structural and the functional unit of life. Thank you.